name's Jackson Blint, and I'm a junior at Holy Trinity Catholic High School in Fort Madison, Iowa. And for my project this year, I wanted to know if I could alter the genetic makeup of E. coli so that way it could survive on ampicillin. What got me into this project was the whole COVID thing in general. I really wanted to learn about viruses and bacteria and how they operate, so I figured I would do a project on that this year. Obviously, I can't solve COVID by myself, but I should start somewhere, so I started with E. coli. When I dove into this project, I learned that sanitation is huge. It's almost half the project. In this project, I really wanted to focus on my lab technique and my sanitation skills. So to start the project, before I did anything, is I sanitized the area in my hands with isopropyl alcohol to make sure everything was completely free of germs so no contamination could happen. Then, to make sure all of my lab tools were sanitized, we would put them in an oven for different temperatures and different times depending on the material. Glass stayed in longer and for a hotter temperature, while plastic was at a lower temperature but even longer, so that way they could all sanitize. Because we can't put stuff together because plastic will melt if we did it at the same temperature that we did glass. But after everything was sanitized, we started with getting the agar mixture ready and the nutrient broth. The agar mixture was relatively simple. We sterilized distilled water by boiling it for 30 minutes, and then we used that to mix into our agar and our nutrient broth so we could get the agar ready to be poured. After everything was mixed and ready to be poured, I set up the plates along the edge of the table so I was ready to pour them without any contamination or fear of it. One thing that was helpful with COVID, which is something you don't hear a lot, is that the mask actually helped so I wasn't breathing directly on the plates, so it prevented a lot of contamination that could have occurred. Usually I found that you would breathe to your side and hold your breath as you were going, but with the mask it helped a lot to prevent a lot of that contamination. After the plates were poured, that's when the real project decided to set in. We got the E. coli ready with the tube that came to us. We used that tube as the starter colony. So what we did was we got the E. coli put into microfringe tubes to get ready for us to use the calcium chloride on them to make sure we could make the cell membrane permeable with the new DNA. The calcium chloride makes the cell porous and have holes in it, so that way I can actually insert the new gene in there without it just getting bounced off and thus not used by the cell membrane. After that was done, we let it sit in the calcium chloride mixture for about 10-15 minutes, so that way we were sure it was ready. That's when some of them got the new DNA added. The B1s got their DNA added for this specific experiment. After the DNA was added, we put them in a warm bath for about 90 seconds so that way they could recuperate, so that way they didn't just die, because the calcium chloride is actually really hard on the cell itself. After that, we put it into an ice bath for about 15 minutes so that way it could recuperate a little bit more. And after that, they went into the incubator for about 20 minutes so that way they could sit there and they could just regenerate and not be so fragile when we transferred them. After we were ready to transfer them to the plates, we did the respective ones, the B1s into the B1s and the B2s into the B2 plates, so that way we could add them. We let them grow for one day, and then we came back and added the nutrient broth to make sure they were helped to be grown. After that, it was just a matter of waiting. We just waited for the results, and as they came out, my hypothesis was supported, and we were able to alter the E. coli to grow on the ampicillin. Thank you for listening to my project.